हेलो स्टूडेंट्स, वेलकम बैक। हम लोग 12th का ह्यूमन हेल्थ एंड डिसीजेस ये चैप्टर डिस्कस कर रहे हैं। उसमें आज मैं बहुत ही स्पेशल ऐसे टॉपिक को लेके आया हूँ एंड द टॉपिक इज HIV इन्फेक्शन, दैट इज एड्स इन्फेक्शन। HIV इन्फेक्शंस और एड्स इन्फेक्शन. What is HIV and what is AIDS? So we will discuss it in detail with these some of the points like the introduction to HIV, transmission of HIV, high risk in HIV, life cycle of HIV, diagnosis, how can we diagnose the disease, treatment of the HIV, prevention and the facts. So after reviving all these points, you will be able to understand the detail in detail what is HIV and how it can be diagnosed and prevented. Okay, so let's start with the transmission of HIV introduction. So very first, what is the long form this HIV and AIDS? So HIV, it means human, I for immunodeficiency virus. Okay, similarly, so human immunodeficiency virus. Second one is AIDS. It is autoimmune it is not autoimmune, it is acquired immunodeficiency virus. Okay, it is acquired immunodeficiency syndrome. So each word, when we discuss and study each word of this AIDS, we will understand what is the disease. Very first, acquired. What do you mean by acquired? As I told you before, that some are acquired and some are innate. Acquired means during life. After birth. It is not since birth. It can be acquired, it can take place after the birth. So it is acquired. Then immunodeficiency. Immunodeficiency meaning, it means there is the decreased or less immunity, or simply we can say weak immunity. Why there is a weak immunity? We will see it discuss in detail. But weak immunity, it means the person is very much susceptible, where can get diseased, can get infected very easily. So immunity is very weak in the disease of AIDS. And last one, it is the syndrome. What does the syndrome mean? Syndrome means it is a group of signs. Group of signs what a doctor can observe. Group of some symptoms what patient says, like patient feels weakness, patient is having giddiness, whatever symptoms, signs. Along with that, physical and also physiological, in body, physiological and physical disturbances. These are the disturbances that are produced in a syndrome. Having these all are taking place because of single cause. There is a single cause. And with this single cause, person is having some signs, some symptoms, some physiological and physical disturbances. So this is what this AIDS is. It means the decrease or less or weak immunity of a person due to a single cause, causing many signs, symptoms, some disturbances related with physical and physiological. So this is what the picture of HIV seems to be. Clear? So this is the introduction. Simply in HIV or AIDS, the immunity of a person get decreased, it becomes weaker, and person is very much susceptible, very much prone, can easily get infected. Clear? Next. Now we will discuss about the transmission of this HIV virus. So human immunodeficiency virus. Okay. So how is the transmission? When we see the transmission of this HIV, so it is through, we can say simply, it is through body fluids. It is through body fluids like blood, lymph, etc. Blood, lymph, etc. These are all the fluids with the help of which HIV get transmitted. Along with that, this transmission is, we can say this transmission is through unprotected sex with infected person. This is the main cause, infected person. Another one thing is unprotected sex with multiple partner. Unprotected sex with multiple partners. This is also a cause. Again, how this can get transmitted? Through blood so unsafe blood transfusion. Unsafe blood transfusion. Also with contaminated, which are contaminated needles and syringes. And the last one, the very unavoidable cause, is through placenta. Through placenta from infected mother to child. This is how this transmission takes place. Through the body fluids, mainly blood and lymph, plasma we can say, unprotected sex with infected person, unprotected sex with multiple partners, unsafe blood transfusion, that is BT. किसी को ब्लड देना बाहर से, अगर वो ब्लड इंफेक्टेड है, अगर उसको क्रॉस चेक नहीं किया तो भी ये HIV हो सकता है. कंटामिनेटेड नीडल्स और सिरिंजेस का बार बार यूज करने के कारण, और लास्ट में कोई मदर अगर इंफेक्टेड है तो उसके प्लासेंटा थ्रू भी ये HIV स्प्रेड हो सकता है. दिस इज हाउ द ट्रांसमिशन टेक्स प्लेस. क्लियर? नाउ नेक्स्ट बिट इज हाई रिस्क. पेशेंट्स ऑन हाई रिस्क. Which patients on high risk? In this high risk, the patients who have sexual contact with multiple partners. Sexual contact with multiple partners. This is the main cause. The patients having intravenous drugs administration. We can say drug addicts or drug abuse. Drug abuse people, these are more prone, so they are on high risk of this HIV infection. Clear? Next is life cycle. This is very important. How this in HIV or AIDS get infected and survives in our body, how it get multiplied. So we can say life cycle. Very first thing, what will happen if there is suppose unprotected sex and there will be the entry of virus. After the entry of virus, it attacks macrophages. It attacks macrophages and it infects RNA. This infected RNA get transformed into infected DNA, infected DNA, and this infected DNA here, this RNA, it replicates, and this replication is with the help of an enzyme, and enzyme is reverse, very important, reverse transcriptase. This infected DNA, next it forms many pathogenic RNA copies. So many RNA copies are produced. This is how the life cycle of this HIV virus into the cell goes on. Second one thing is that here it enters in T lymphocytes. T lymphocyte, it means there is a cellular immunity. This T lymphocytes, it means T lymphocytes means helper T lymphocytes. These helper T lymphocytes, they get infected, and then there is the damage of T lymphocytes. T lymphocytes, it means decreased T lymphocytes, helper T lymphocytes, and there is decreased cellular immunity. It means patient is again very much prone to the infection. Clear? How is the life cycle of the HIV into the human body? So entry of the virus. After the entry of virus, it attacks the macrophages. So two cells get destroyed and damaged, macrophages, and another one is helper T cells. After that, these RNA get infected, it gets replicated, and then it forms infected DNA, which multiplies the copies of infected and pathogenic RNA. Along with that, T lymphocytes also get hampered, and number of T lymphocytes get decreased, and as a consequence of it, there is less cellular immunity, and patient is again get prone to this infection. Okay, so this is how the life cycle of HIV takes place in human body. Next bit is diagnosis. How can you diagnose the patient? So next one is diagnosis. Diagnosis is with ELISA. ELISA means enzyme linked immunosorbent, and last one is assay. This is very important. ELISA test, it is used for HIV. Vidal test, it is used for typhoid. Okay, so ELISA is the test by which we can diagnose the HIV or AIDS. Okay, next is treatment. Simple. What is the treatment of this HIV? It is very simple. Why I am saying very simple? Because in treatment, very simple, no medicines are available for treating the AIDS. So what is the best treatment for this HIV? The best treatment is very simple, prevention. So that's why it is said prevention is always better than cure. So prevent yourself from getting infected. This is the best care and cure for the HIV. Next is prevention. Then how can you take prevention? Prevention, very simple, avoid sex with many partners. That's why it has to be educate the people. Second one thing, distribution of free condoms to the human beings. Another one thing is using safe and sterile needles and syringes. Person who needs multiple times blood transfusion, he is more prone, he has more chances of getting infected. That's why To prevent it, very simple thing is educate the people about the AIDS, how it get transmitted. Avoid sex with multiple partner. Use free condom so that people can use it who are not able to buy that. Then another one is safe and sterile needles and syringes use. Another one thing what we can do is the disposable syringes. Disposable and use and throw syringes and masks. Syringes and needles. That we can do to prevent the HIV. Okay, and last one thing is that facts. How this? Because in our society, even today, many of the people are not aware of the HIV. They don't know. They are not educated about the HIV. That how it get infected, how it get transmitted. So facts. What are the facts about HIV? So facts are that not spreaded by, and another one is spreaded by. How it gets spreaded, how it doesn't get spreaded. So not spreaded by direct body contact with the infected person, not by sneezing, not by insect biting, not by being together, not by sharing clothing, not by sharing some we can say game instruments like bat, ball, rackets and everything, not by touching somebody's handkerchief, not by sharing plates. These are not spreaded by all these means. Then how it is spreaded? It is spreaded through body fluids, not even swimming pools, not even through swimming pools, not using we can say community toilets, socially using toilets, socially using telephones, all are not the means of spreading. So these are spreaded by body fluids. These are spreaded by BT, blood transfusion. We can say contaminated. We can say contaminated and non-cross-checked, and infected blood. So this is how is the picture of HIV and AIDS. AIDS similarly, if person get infected by HIV, a simple ulcer or simple rash on a skin can cause a large ulcer. Simple infection can cause diarrhea. A simple cold and fever can cause severe pneumonia. So patient gets infected by all these things and patient dies finally with not because of HIV infection directly, but due to the severity and the diseases produced, and we can say the associated complaints, associated diseases, associated infections, they are capable of dying the person. So I hope you might have understand the HIV and you might get a good message. You can spread this message to the society that how we can spread getting infected and how we can treat the patient, how we can spread this message to the society. So keep studying. Thank you so much.